Hello, everyone. Welcome to the science behind evidence because evidence doesn't lie. Let's learn how to read it. Today, we are exploring blood, its composition, functions, and forensic significance. We'll be covering this in two parts. First, the biology of blood, that is its composition, functions, and all. And in the second part, we'll do forensic identification. In that, we'll be doing your presumptive test and confirmatory test for blood. Let's start with what is blood. So, blood is a fluid connective tissue composed of plasma, blood cells, and platelets. It circulates continuously through the cardiovascular system. It transports oxygen, nutrients, hormones, and waste products, and it plays a central role in immunity, clotting, and thermoregulation. It covers 8% of your total body weight and 5 to 6 liters average volume in an adult. And now, first of all, we say it is a fluid connective tissue. But why do we call it a connective tissue when it's a liquid? The answer to this is most people imagine connective tissue are something solid like bone or cartilage. Blood is classified as connective tissue because it originates from the mesoderm and has cells suspended in an extracellular matrix, that is plasma. It connects different organs by transporting oxygen, nutrients, hormones, and immune cells throughout the body. And let me tell you an interesting fact about blood. If all the blood vessels in an adult were placed end to end, they would stretch for approximately 100,000 kilometers. That is enough to circle the earth more than twice. Now, coming back to the components of whole blood. When you centrifuge the blood, it separates into distinct layers that are plasma, erythrocytes, and buffy coat. Plasma being the most, 55%, erythrocytes, that is your RBCs, approximately 45%, and buffy coat, that is leukocytes and platelets. They constitute less than 1%. Now, the question here is why does blood separate into three layers after centrifugation? Because every component has a different density. RBCs are the heaviest because they contain large amounts of hemoglobin. Plasma is mostly water, making it the lightest. WBCs and platelets have intermediate density, so they form the buffy coat. Now, after centrifugation, the erythrocytes take up the bottom layer because they are the heaviest. Plasma takes the uppermost layer because it is the lightest, and the buffy coat stays in between. It is the intermediate layer. Now, this buffy coat is often used for DNA extraction because leukocytes contain nuclei while mature RBCs do not. Next, we have the classification of blood constituents. The whole blood is divided into plasma being 55% and formed elements being 45%. Plasma contains water, nutrients, and electrolytes, amino acids, and nitrogenous wastes, gases, and proteins, whereas formed elements contain erythrocytes, leukocytes, and platelets. Leukocytes also are divided into granulocytes and agranulocytes. And the granulocytes are further divided into neutrophils, eosinophils, and basophils. Agranulocytes, on the other hand, are divided into lymphocytes and monocytes. Next comes plasma. Plasma is a pale yellow liquid portion of blood that makes up 55% of total blood volume. Now, why don't we simply call it water? Plasma contains far more than water. It contains nutrients, antibodies, hormones, clotting factors, electrolytes, proteins. Water is simply the solvent. Okay, the important proteins of plasma are fibrinogen, albumins, and globulins. It transports nitrogenous waste and dissolved gases. Liquid state of blood is largely contributed by plasma. I have also written its clinical relevance. Plasma transfusions are given to patients with liver failures and also used for patients with life-threatening injuries and severe blood loss. Next comes erythrocytes, that is the red blood cells. Before that, I just wanted to tell you something important, that is about serum. If fibrinogen is removed from the plasma, what remains is serum. I think students often confuse the two terms. The easy trick for that is plasma is called when clotting factors are present. Serum, it is when the clotting factors are absent. Okay. So now coming back to erythrocytes, the red blood cells. They are biconcave shaped cells that lack a nucleus in humans. Now, why don't mature human RBCs have a nucleus? Removing the nucleus creates more space for hemoglobin, allowing RBCs to transport more oxygen. However, without a nucleus, they cannot divide or repair themselves, which explains why their lifespan is limited to around 120 days. Another question is, why are RBCs biconcave? Because this shape increases surface area, speeds oxygen diffusion, and makes RBCs flexible enough to squeeze through tiny capillaries. An interesting fact is, an adult produces approximately 2 million RBCs every second, and they contain iron-rich proteins, hemoglobin, which gives blood the red color. This is why the white blood cells are colorless, because they do not have this hemoglobin. This is responsible, hemoglobin, for the transportation of oxygen. They are produced in the bone marrow. I think there is a mistake. This is not 3 months, this is 4 months. Okay. Moving to hemoglobin structure. Hemoglobin has 4 polypeptide chains, 2 alpha plus 2 beta chains. Each chain carries a heme moiety that binds oxygen. Heme is known as protoporphyrin 9 plus a ferrous ion, and together they are called as ferroprotoporphyrin. This is the structure for hemoglobin. These are the 2 alpha and 2 beta chains, and we have space here. I'll write. HbA is for major adult hemoglobin. Sorry for this writing. It has 2 alpha and 2 beta chains. Then we have HbA2. It is for, that was major adult hemoglobin. This is minor adult hemoglobin. It has 2 alpha and 2 delta chains. Next we have is HbF, which is for fetal hemoglobin. It has 2 alpha and 2 gamma chains. Okay, now going to leukocytes. Leukocytes are your WBCs. They are colorless blood cells. They do not contain hemoglobin, mainly contribute to immunity and the body's defense mechanism. They are classified broadly into 2 groups. based on the cytoplasmic granules. Granulocytes that contains granules in their cytoplasm, and agranulocytes that do not contain granules in their cytoplasm. Granulocytes are divided into neutrophils, eosinophils, and basophils, whereas agranulocytes are divided into lymphocytes and monocytes. Firstly, we'll talk about granulocytes. In that, we have eosinophils. They make up 2 to 3% of leukocytes. They are produced in the bone marrow, found in high concentration in the digestive tract. They are responsible for combating parasitic infections of vertebrates and are involved in controlling mechanisms associated with allergy and asthma. This is why doctors say during any allergic reactions, the eosinophils increase in number. The key point is eosinophils are the body's main defense against parasitic invaders and play a central role in the allergic reactions. Next is basophils. They constitute 0.5 to 1% of total leukocytes. They contain large cytoplasmic granules that play a vital role in mounting a non-specific immune response to pathogens. They release histamine, which triggers an allergic reaction and dilates blood vessels. They help maintain the thickness and consistency of blood, stain readily when exposed to basic dyes, hence their name is basophil. They are best known for their role in asthma and inflammation and bronchoconstriction in the airways. They secrete serotonin, histamine, and heparin. Basophils mediated histamine release is relevant to understanding injury response timelines in forensic medicine. Next, we have neutrophils. They are most abundant WBCs, first cells of immune system to respond to an invader such as bacteria or virus. They constitute 60 to 65% of leukocytes. Their diameter is 10 to 12 micrometers, nucleus is 2 to 5 lobed, cytoplasm has very fine granules. They help destroy bacteria using lysosomes, act as strong oxidants, stained only using natural dyes, normally found in blood streams, predominant cells found in pus. They circulate for 6 to 10 hours before migrating into tissues, where they survive for 1 to 2 days. They are produced every day in the bone marrow. Forensic significance here is neutrophils flood an injury site rapidly. Their presence and depletion into pus helps estimate whether a wound occurred before or after death. This is because their presence in the pus cells, and pus is formed when the body is in the healing stage. When your injury is healing, there is this formation of pus, and the presence of neutrophils shows the antemortem injury. This is not a sign of postmortem injury. And in fact, the pus is composed largely of dead neutrophils, dead bacteria, and the tissue debris. Now, moving on to monocytes, the agranulocytes. They make up 6 to 8% of leukocytes. They are large, bilobed nucleus. No, the nucleus is not bilobed. They generally have half moon or kidney shaped. Their diameter is 10 to 20 micrometers. They are known as the garbage trucks of immune systems. I forgot to mention one more point. The neutrophils can even release the neutrophil extracellular traps. The NETs, neutrophils extracellular traps. These are DNA-like web that trap the bacteria. Now coming back to monocytes, they are known as the garbage trucks of immune system. They clean up dead cells and protect against blood-borne pathogens. They move very quickly to site of infection and tissues, and they remove the dead cells, the dead bacteria, the damaged tissue before that healing begins. They are single bean-shaped nucleus, hence they are known for their role in phagocytosis. Phagocytosis is monocytes engulf and digest the pathogens and cellular debris to keep the tissues clean, hence the name garbage trucks. And also, after entering the tissues, monocytes become macrophages. Next is lymphocytes. They constitute 20 to 40%. They play a vital role in producing antibodies and body defense. Size ranges from about 8 to 10 micrometers. They are colorless cells formed in lymphoid tissues, hence referred to as lymphocytes. They are responsible for humoral and cell-mediated immunity. The three major types, each with a distinct role, are B cells, T cells, and NK cells. B cells make antibodies to fight germs. T cells directly attack specific infected areas. NK cells are the natural killer cells. They destroy viruses in cancer cells naturally without needing prior exposure. Then lastly, we have platelets. They are specialized blood cells produced from the bone marrow. When there is bleeding or hemorrhage, platelets help in clotting and coagulation of blood. They make up less than 1% of whole blood alongside leukocytes. That's the buffy coat. And why it matters is platelets are the body's first responders at a wound site. Their rapid aggregation and clot formation prevent excessive blood loss and initiate the healing cascade. Why are platelet called cells if they do not have the nuclei? You know that. This is because they are cell fragments produced from giant bone marrow cells that are called megakaryocytes. An interesting fact about platelets is platelets release growth factors that help wounds heal after clotting. Then we have the next part that we'll cover in the next video. That's all for today's lesson. If you found this helpful, share it with your friends who loves forensic science. And remember, evidence doesn't lie. It only waits for someone who knows how to read it. I'll see you in the next episode of The Science Behind Evidence. Till then, take care. Bye bye.